Great.
Hello, everyone, and welcome to today's webinar for mental health professionals, Genetics of Depression and Anxiety, What Do Clinicians Need to Know, featuring Jordan Smoller. My name is Astrid Masfar. I am the Senior Education and Outreach Coordinator here at ADAA. Before we get started, I would like to go over a few items so you know how to participate in today's webinar. We've taken a screenshot of an example of the attendee interface. You should see something that looks like this on your own computer desktop in the upper right corner. You have joined the presentation listening using your computer speaker system by default. If you'd prefer to join over the telephone, just select telephone in the audio panel and the dial-in information will be displayed. You will have the opportunity to submit text questions at any time during the presentation by typing your questions into the chat box in the control panel. You may send in your questions at any time and we'll collect these and address them during the Q&A session at the end. If you registered and paid for CE credit, you will need to provide the 4-letter CE code in the webinar evaluation. The CE code will be shared at the end of the presentation, and you will receive a follow-up email including the webinar evaluation, webinar recording, and post-test assessment for those seeking CE credit. You will also have the opportunity to complete the post-test assessment after the presentation. The questions will appear on your screen, and you will be given 5 minutes to complete the assessment. Once complete, please click Submit. I would now like to introduce Dr. Jordan Smoller. Dr. Jordan Smoller is the MGH Trustee's Endowed Chair in Psychiatric Neuroscience, Professor of Psychiatry at Harvard Medical School, and Professor in the Department of Epidemiology at the Harvard School of Public Health in Boston. He is Associate Chief for Research in the MGH Department of Psychiatry and Director of the Psychiatric and Neurodevelopmental Genetics Unit in the MGH Center for Genomic Medicine. I'm now going to take a moment to pass the screen to our expert. There will be a brief pause while I do so.
Can you see my screen?
No, not yet.
Okay. I'm trying to click on the button that allows me to show my screen, but my cursor is not visible here. Hold on one second.
Okay.
Okay, you want to try that again, possibly?
Sure. All right. Let me know if you received the invite.
Got it. Do you see my screen?
Yes.
Fantastic. Okay. Thank you, and thanks to everybody for joining today. It's a pleasure to try to give you a little bit of an update about what we know about the genetic basis of depression and anxiety and what clinicians need to know at this point. And so I'm going to give you a little bit of an overview of what's been going on in genetic research in the last number of years and talk a little bit about what the future may hold. And I will try to minimize some of the arcane technicalities and really focus on some important takeaways. So why don't we get started? For some reason, my screen is not advancing. There we go. Okay. The first thing is whether and how we think about the importance of genetics in clinical practice. And we actually got interested in this nearly 15 years or so ago now, because we had the sense that genetic information was going to become much more relevant to clinical practice in the future. And we actually did a survey at that time of psychiatrists' knowledge and opinions and practice patterns, comparing them to genetic counselors and also internists. And there were a number of interesting findings that emerged from that. But I want to focus on one in particular, and that had to do with how prepared people felt to talk about genetic information with patients, clients, and families. So we asked people, do you feel competent to discuss genetic information regarding, it says psychic illness, but psychiatric illness, let's say, with patients and their families? When we asked that question, Most people said no, they did not feel competent to do that. When we asked similarly if their medical training had prepared them to do that, again, most people said no. And then when we asked, is it your role to discuss genetic information regarding psychiatric illness with patients and their families? 80% said yes. So the upshot was that people felt like this was within the scope of what they should be doing clinically, but really felt unprepared to do it. And so one of the reasons that I think it's important for us to talk about what's going on in genetics is to be able to answer questions, to be able to know when we don't have the answers to questions, and how to begin to talk about this in clinical settings. Here's another question that emerged from a study more recently, in which patients who were affected with psychotic illness and their family members were asked about what they think the risk of the condition was in various scenarios. So scenario A was, what do you think is the chance, excuse me, that someone who has no family members with psychosis would develop psychosis themselves? And they were given these choices: 1%, 10%, 25%, 50%, or 100%. And then, what do you think is the chance for someone who has a brother or sister with psychosis to develop psychosis themselves? So think about that for a moment, how you would answer that. And then I'll show you that what people said was that there was a fair bit of actually variability. Most people in scenario A thought that the risk was about 1%. That is, there's a 1% chance that somebody without any first-degree family members would develop psychosis. And that actually is the answer. But a substantial proportion of people felt otherwise. And then in scenario B, the most common answer was, well, it depended on who you asked, but for relatives, the most common answer was 10%. And for affected individuals, the most common answer was actually much higher. But the correct answer is in bold there. It is 10%. So there's a lot of uncertainty about what risks are involved, how much genes contribute. So let's talk a little bit about that. We're going to focus, of course, on depression and anxiety, but I want to make a few takeaway points that we'll go through as we go along. Some of these are generally speaking true of psychiatric disorders. So the first takeaway is that all psychiatric disorders have a familial and heritable component. The second is that we've begun to actually identify the genes that contribute. The third is that depression and anxiety disorders are what we call polygenic, and I'll explain what that means. And the fourth, that genetic influences cross our clinical diagnostic boundaries. We'll talk a little bit about clinical implications now in 2018 in terms of genetic testing and pharmacogenetics. And the takeaways there are that the genetic testing applications are really pretty limited at this point. In terms of pharmacogenetics, which has to do with, can we use genetic information to guide treatment? This is a really important area, as we'll talk about, but the evidence still is limited. And then we'll talk about what the future might hold. So that's what I'm going to try to cover today. And we'll start with the notion of how influential genes may be in psychiatric disorders. When you approach genetic research, there's really a series of questions we ask and a series of types of studies we use to answer those questions. And I'm just laying this out for you because we will return to some of these. The first question is, does the disorder run in families? And we can get a relative risk, essentially, what we call the recurrence risk ratio, for how much more common it is in the relatives of first degree relatives of affected individuals compared to the general population. If it runs in families, of course, that doesn't necessarily mean that that's because of genetic variation, because things like environments and experiences also run in families. And so we typically turn to things like twin studies to sort of try to tease apart how influential are genetics in that kind of familial risk. And then if we think that's the case, that genes do influence the risk, we have in the last couple of decades, really more than nearly now 30 or more years, been able to begin to look at the DNA itself. And there are a whole series of study designs. I won't go into great detail on these, but they're asking questions like which chromosomes are involved, where are the genes that might be involved, and what specific genetic variants might be involved. And then people are very interested in how genes and environment interact. And that's a whole area, which is still in its very early stages. And all of that is in many ways a prelude. It's sort of the first steps to actually figuring out how you get from genetics to a clinical condition. What are the mechanisms? What's actually going on? And there are a whole variety of studies that combine molecular biology and neuroimaging and cellular studies that are now possible to study genes that emerge from these genetic studies. But let's go back to this first question about how influential genes may be. So the first takeaway was depression and anxiety disorders have a familial and heritable component. So look at this list of conditions and think in your mind about which of these you think might have the strongest heritable component, where genetic differences among people account for the most proportion, the greatest proportion of risk in the population. I'll give you a moment to look at that list. And then I'll tell you that from twin studies, which again allow us to estimate this concept of heritability, which is the proportion of risk in the population that's attributable to individual differences in genetics, this is what the numbers look like. So autism, bipolar disorder, schizophrenia, ADHD are all quite highly heritable. Then there's a tier of kind of mid-heritable disorders, alcoholism, eating disorders, depression, and anxiety disorders. And then you notice that Parkinson's disease and breast cancer are actually the lowest on this list, even though genes for those disorders, especially early onset versions of those disorders, were identified a number of years ago and are highly penetrant, as we would call it, meaning if you inherit one of those genes like BRCA1 for breast cancer, there's a pretty high probability that you might be affected. But the heritability is really just the overall contribution of genes. And so it may be that something like ADHD, which doesn't have a big single gene involved, nevertheless has the added effects of many thousands of genetic variants that add up to a large heritability, whereas breast cancer, despite the fact that there are these highly penetrant genes, most of the cases of breast cancer are not due to those genes, and so their contribution to the overall heritability may be more limited. But the clear takeaway here is that psychiatric disorders are highly heritable. Genes clearly play a role, and depression and anxiety disorders are moderately heritable, we would say, from these twin data. I mentioned that the other way to look at this is in terms of familial risks. And this is a table that shows you what we would call the recurrence risk ratio, or the first degree relative risk, you could think of it as, which is how many times more likely are you to be affected if you are the first degree relative, that is, let's say, the sibling or child of somebody with the condition, compared to somebody in the general population. And you can see again, for all of these conditions, there is an increased familial risk. Anything greater than 1 times means there's an increased risk. So for things like depression, there's a 3-fold increased risk over the general population. For anxiety disorders like panic and phobic disorders, maybe a 5-fold. And for something like schizophrenia or bipolar disorder, maybe a 10-fold increased risk. So we've established that these are heritable familial conditions. Now we want to begin to actually identify what genes may be contributing. And the takeaway here is that we've begun to really have some success there. Just to remind everybody what we're talking about when we talk about genes, every cell in your body, except for a couple of minor exceptions, has 23 pairs of chromosomes. And those chromosomes are long strands of DNA. And the DNA is made up of sequences of bases, A's, T's, C's, and G's. And those bases are organized into units that we call genes. And the genes really are making proteins that are crucial to running the cellular functions in our body. There are other things that the DNA codes for besides protein, including factors that regulate other genes. But for the most part, we can sort of think in this somewhat simplified way. So what we're actually asking is, can we identify which specific genes or variants of those genes might be involved in the disorders that we're interested in? And we've seen a lot of progress. The way that people have typically looked at this is to study two kinds of DNA variation, especially in psychiatry. The first kind is what we refer to as a SNP, or single nucleotide polymorphism. So if you think of the text of the DNA as these strings of A's and T's and C's and G's, substituting one letter difference between people is a single nucleotide polymorphism. Now, there's another kind of variation that has also proved to be important in psychiatric illness, and that is copy number variations. These are much larger changes in the DNA. They are deletions or duplications of chunks of DNA on a chromosome that may involve 500,000 of these letters at a time, or 1,000,000 of them. And it turns out that we all carry varying degrees of these as well. And some of them, especially ones that are very rare, have now been shown to be related to certain psychiatric disorders. So genetic studies in general are asking which DNA variations are more common in those with a condition compared to those without a condition. So it's a fairly straightforward kind of question we're asking. And we began asking that question maybe 25 years ago and didn't really have much progress until recently. So there was almost nothing that had been kind of convincingly identified as associated with a psychiatric disorder until about a decade ago. And what happened then was that there were major advances in technology and also in collaboration, scientists coming together and sharing data. And we were able to look across the entire genome at once, rather than picking and choosing spots on the genome that we thought might be involved. And that was partly the way we proceeded because of technical limitations. But the bottom line is that in that last decade, with ever larger studies, we have seen the identification of very strongly associated, statistically strongly associated variants for a variety of conditions. Just this last week, there was the report of new loci found for, or DNA variants found for ADHD, autism, bipolar disorder. You can see depression, schizophrenia, now with maybe 250 or 300 or more variants that have been convincingly associated. Post-traumatic stress disorder, a little bit lower on the list, but still yielding findings. So this has really been a pretty dramatic change over the last decade, and one that has opened up all kinds of new windows. I want to just give you a sense of what we've seen in depression, for example. And this is a recent paper in the journal Nature Genetics, which kind of illustrates a point about what we're finding in genetics. So this was a study, a collaboration by the Psychiatric Genomics Consortium, which is a worldwide consortium of scientists who put together a very large study. You can see there are about 135,000 cases and 345,000 controls. And again, what we're asking is which variants on the genome are more common in the cases than the controls. And the figure that you're looking at here is how we depict in one single picture the results of a genome-wide study where we've measured markers across the genome. We call this a Manhattan plot. And you can maybe surmise why it got that name. There are lots of skyscrapers, at least we hope to see skyscrapers. What you see on the bottom of a plot like this are the numbers 1 to 22, which are the first 22 chromosomes. The 23rd pair is the sex chromosomes, which are not shown here. And each of the little dots that you see are markers lined up according to their position on the chromosomes that have been tested for whether they are more common in cases than controls, in this case, depression cases than controls. And you test about 1 million of these markers all at once. So you're looking at a picture that has 1 million markers or statistical tests. And the red line that you see across there is our threshold for statistical significance, which is very stringent. So the y-axis going up and down is the degree of statistical significance. And so a 6 there, let's say, on the y-axis means a p-value that we use to establish significance of 10 to the minus 6. So we're asking for a very, very low p-value, very stringent. Anything above that line is a region where we see significant association. So you can see that these common SNPs, these common variations, some of them are shooting well above that line. And in fact, there were 44 that really made it clearly above our statistical threshold. So that's how we kind of evaluate and do one of these studies. I mentioned these copy number variations, and in another recent report, it turned out that certain deletions or duplications of the genome were also associated with depression. And what you're looking at here is a figure from this paper, which is currently in a preprint form. And what you see on the y-axis there are just the names of copy number variations. So you may see 1q211.1 del. What that means is chromosome 1, the long arm, and then there's a band location, 21.1, so that's where it's located, and it's a deletion of that. And you can see that there are a bunch of these that pop up, again, beyond our threshold for statistical significance in this case, which is different. But the odds ratios, the effect size, is larger typically for these, the ones that have been found. So I won't go into great detail here, but just to say that common SNP variations and rare copy number variations have been associated with psychiatric disorders and, in this case, depression. Now, just getting a list of these things is amazing and interesting, but it doesn't by itself tell us that much about the story. What you really want to know is what's going on biologically. And in the last few years, what we've seen is that many of these variants and genes that are implicated start to converge on biological pathways. They start to tell us stories about what's going on at the biological level. And I won't try to explain the graph on the left there, but basically it's a way of visualizing which biological pathways are most important. And many of these have to do with the function of neurons, for example, which maybe is not so unexpected. But the major point to keep in mind here is simply that we are finding pieces of puzzles that we are now being able to put together to get a better sense of what's the underlying biology. And what that means is that every time we get a variant and particularly a pathway, we have a clue for a new target that might be useful for developing effective treatments. And that's really important because, as you undoubtedly know, in terms of FDA-approved treatments for depression and anxiety, they are all based on biological hypotheses, mainly monoamine transporter genes, serotonin reuptake inhibitors, and tricyclics and MAO inhibitors. They're all based on the same biological idea from the 1960s, essentially. This is actually telling us things we didn't know before and pointing us in new directions. And so many people are now trying to go from these confirmed genetic associations to figure out what they're doing at a biological level, to then screen all kinds of potential compounds to develop drugs that are actually targeted to the biology. And so what this is just a kind of a flow chart for what's involved in that. It is a bit of a shortcut in the sense that we're tying it to the biology, but it's by no means easy. But we are beginning to see in fields like cardiology and cancer or oncology exactly this kind of thing working well. And the hope is that we can bring that to the treatment of neuropsychiatric illness as well. Third takeaway is that depression and anxiety disorders are highly polygenic. And this is related to something that I was just talking about. There is no the gene for a psychiatric disorder. So if you hear people say, you know, the gene for depression was found, that doesn't really mean anything. It's not a single gene. The genetic component of these conditions, like schizophrenia and other psychiatric conditions, is made up of many genes and DNA variations. Some of them, these SNPs, are common. That is, we all carry them to varying degrees, and they have small effects on their own. And some are rare and may have larger effects. Some are inherited. Some may be de novo, as we call it. What that means is that they arise in the embryo or in early development and were not passed on from the parents, particularly these rare de novo copy number variations. The common SNP part, again, typically there are thousands and thousands of these that may contribute, but on their own, they have effect sizes. Their contribution is tiny. It's just that there are thousands of them and they add up all together to a much larger effect. People have been very interested in doing that, in trying to estimate somebody's genetic risk by adding up all the individual variations into a score, a genetic risk score, sometimes called a polygenic risk score. And basically what you're doing there is you're adding up all of the individual effects of the variants that somebody carries into a single number. And those effects, when you add them all together, tend to be much greater, obviously, than the individual single variant effects, but they still only account for a minority of the genetic component of things like depression. Okay, the fourth takeaway is gene effects cross our clinical diagnostic boundaries. People often like to say that our DNA has not read the DSM. So we know that our system for defining disorders largely has been based on the DSM, which has evolved since 1952 to the latest edition in 2013. And what you're looking at here is the number of diagnostic labels in the book. So you see immediately, and I think this is no surprise to anybody, that our clinical diagnostic boundaries and definitions are not exactly stable. And we may not exactly know where the optimal place is to define a disorder and place a line. And in fact, if you just look at the evolution of things like autism-related disorders, mood disorders, and anxiety disorders from DSM-1, you're seeing a timeline here moving it up to DSM-3 to DSM-4. And what you notice is that new disorders are coming in and old disorders are dropping out. And this is just 3 categories of disorders. In DSM-5, we actually added some new categories, but we also reduced autism to a single overarching category. Just making the point that the boundaries of our diagnostic system are really not all that well defined. And perhaps it's not surprising then that the genetic findings that we have don't map purely onto this kind of a system. The largest look at this came out earlier this year using genetics to estimate how related are different conditions. And this was an effort by what's called the Brainstorm Consortium. And it actually used DNA data for 23 different brain-based conditions, some of which are largely treated in psychiatry, some in neurology. And what was done here was to take all of the genetic variation using DNA and look at how correlated is the genetic component to these different disorders. So on the left, in the section that says psychiatry, what you're looking at is the correlation between pairs of disorders that we think of as psychiatric disorders. The darker the blue and the more you see a star inside the box, the more genetically related those conditions are. So obviously, in the upper left-hand corner, ADHD is perfectly genetically related to itself. But notice that ADHD is also genetically overlapping with bipolar disorder, with depression, with schizophrenia. Depression is overlapping also with OCD and with Tourette syndrome, and so on. Then when you look at this in neurology, disorders that have typically been considered neurologic disorders, things like Alzheimer's disease and epilepsies and stroke, really not that much overlap in the categories genetically, except for migraine to some degree. although those are really mainly subtypes of migraine. And now, interestingly, if you put psychiatry versus neurology and look at the overlap, there's again not that much, which is interesting because we tend to think of the biology of brain-based disorders as certainly linked. They're linked by the organ in which the the activity of the disorder we think is located. But at a genetic level, there seems to be not as much overlap as you might expect. Nevertheless, among psychiatric disorders, there's a whole lot of overlap. Genetic testing is very limited. This is my fifth takeaway for you. And the bottom line here is that for the most part, there are no established genetic tests for common mental health conditions, with a couple of exceptions. And the effect of most genetic risk variants is too small to be clinically meaningful, especially on their own. I showed you that in a number of slides earlier on, a number of parts of this talk, that if you test a particular variant, it's not likely to be highly predictive unless it is a rare, large effect variant, but there really aren't very many of those that we know about at this point. Going back to a survey that was sort of interesting, I think, done by a group at Columbia, asking people, psychiatrists, which of the following conditions do you think have genetic tests available? And you can look at that list. And when they surveyed psychiatrists, these were the results. So take a look at that and think about what you would say. Which of these conditions do you think actually has a genetic test available that's, a clinically useful test? Here's the answers. You can do a test for early onset Alzheimer's disease. And there is also another variant, which people may know about, called the APOE4 variant, which increases risk of Alzheimer's disease. Autism, yes, we'll mention in a moment. And schizophrenia, there arguably are some rare copy number variations that we can test for, including one on chromosome 22, which is sometimes called the velocardiofacial syndrome or DeGeorge syndrome. In any case, it's really mainly autism. Mainly autism, for which there is some consensus that clinical genetic evaluation is appropriate. And these are guidelines from the American College of Medical Genetics that concluded that a genetic evaluation should be offered to all persons with autism spectrum disorder or his or her family. And there are a few different levels of tests that can be done, which together can clarify maybe up to 30 or 40% of cases in which we don't necessarily know the cause. We can find a genetic cause in a substantial fraction of those. What about tests to guide treatment? This is obviously something clinically that we're very interested in and that would have tremendous potential. So 1 in 5 Americans are prescribed psychotropic drugs, but as you know, we don't have a great basis for knowing who's going to respond to what medication, who's going to have an intolerable side effect. It's really pretty much trial and error still. And each time we go through one of those trials, we may have 4 to 12 weeks of unremitted illness before we are, able to say whether this treatment worked. And obviously there's a potential for significant adverse drug effects with every trial. People often get discouraged and say, I'm not going to do medicine. This is not going to be helpful to me because this didn't work or I couldn't tolerate it. And there are enormous costs associated with this. Now, we do know that response to antidepressant drugs, for example, is partly heritable. There is a genetic component. And we also know that there are some severe adverse effects that may be influenced by genes, like weight gain, diabetes, in response to things like antipsychotic medications or mood stabilizers, agranulocytosis with clozapine, and so on. So it would be tremendously helpful to have some guidance about what medicine is likely to work or be tolerable for people. And so there are lots of efforts, and there are in fact tests available to look at this. and typically when a genetic test is done for pharmacogenetics, for trying to understand what role genes play in predicting response, there are two classes of genes that get tested. One are what we call pharmacogenetic genes, which are typically involved in drug metabolism, in the liver, for example, or elsewhere, or the gut or the kidney. And the other are pharmacodynamic genes, that is, genes that we think are involved in how the drug works. So, for example, the serotonin transporter is obviously a gene that makes the serotonin transporter protein that we think is pretty important to the role of serotonin reuptake inhibitors. That's the protein that they directly block. So when you think about the pharmacokinetics, you're thinking about things like cytochrome P450, 2D6, many of the enzymes that we think of in clinical practice when we're worried about drug interactions. But these are the kinds of things that are tested. Now, recent large genome-wide association studies, don't worry about the details of the slide. I just want to make the point that even with large-scale genome-wide studies, we haven't found really any strong associations, except that some of these P450 enzymes do clearly affect drug levels for the drugs that are metabolized by them. Some people are poor metabolizers, their genes do not make a protein that works all that well, and some people are extensive or even super metabolizers or ultra rapid metabolizers, and that can affect drug levels and perhaps side effect rates and effectiveness rates. But we don't have a whole lot other than that we can really kind of hang our hats on. That doesn't mean that people haven't tried to do this and to bring this to clinical practice. There are a growing number of commercial tests that are available. Typically, they include some combination of these P450 enzymes and variations in genes that people think might be involved. They may actually be covered by insurance, and there are studies that have looked at whether these things are associated with better outcomes or treatment response, and those are largely retrospective studies where you have people who you know what happened to them, and then you ask, was that associated with what their genotype might have been? But what we don't have are convincing prospective randomized trials that are published at this point. And so I would say that while it is, I have certainly heard stories of folks who have been helped by having this kind of information. It is still unclear when and if the current tests that we have are helpful. Not to say that they're never helpful, just not entirely clear when and what's the best way to make use of that information. There are some trusted sources out there that provide vetted, published, evidence-based evaluations and guidelines, and I've listed those websites here. And so you might, if you're interested in seeing what's the state of our knowledge, go there to sort of look around. Okay, number 6 finally is there are some interesting things on the horizon. Genetics may inform opportunities for what people sometimes are calling precision psychiatry and prevention. The notion of precision psychiatry builds on the concept of precision medicine, which is an emerging, this is the definition from the Precision Medicine Initiative Work Group, sponsored by the NIH, an emerging approach for disease treatment and prevention that takes into account individual variability in genes, environment, and lifestyle. So the notion is if we learn more about the role that genes play, we learn more about the role that environment and experience plays and how lifestyle interacts with that, we may do much better in predicting treatment response, in perhaps even reevaluating our diagnoses, in tailoring prevention to individuals rather than a kind of trial and error, perhaps one size fits all approach that we have sometimes been limited to. Our own research, where I am here at Mass General, we've been very interested in the way that genetics may actually inform how we understand opportunities for enhancing resilience and preventing complications of illness. And so if you think about where we stand with things like prevention, what are the things that we know are useful in, if you wanted to prevent psychiatric illness, what would they be? Well, one is don't have affected first-degree relatives. 2 is avoid significant childhood adversity. Those are 2 known risk factors. And 3 maybe would be just say no to drugs. So these are 3 things that, genetic risk, early adversity, and substance abuse. At least 2 of those are very difficult to do much about. You really can't do anything about them. And the substance abuse is also often, very difficult to minimize. Oops, sorry. Let me go back there. So there are 2 other things when it comes to potentially modifiable resilience factors for depression that people have studied over and over and have had promising results. One of them is physical activity, and the other is social support. And so in a series of studies, we've been looking at, can we validate and see whether these are, in fact, very useful kinds of areas that might help minimize the risk of depression, boost resilience, and can we use genetics to help us in that quest? And I'm going to tell you the results of a couple of studies that have been led by Carmel Choi, who's here. And one of them had to do with whether physical activity is causally related to reduced risk of depression. So what that meant was there's clearly an association between physical activity and depression. And some people have implicated certain biological connections like neurogenesis that might result from physical activity. But if you do an observational study and you ask, is there an association, and you find that there is, it's certainly possible that physical activity could be reducing risk of depression, or that depression could be reducing physical activity because people move around less. What's the causal connection? It turns out that genetic studies now provide us with a new way of testing that. You can't easily, for many things, randomize people and follow them and in large numbers get an answer. Certainly you'd ideally want to do a randomized controlled trial. It turns out that because we inherit our genetic variation in a random fashion, whether you inherit one or another version of a variant is random, if you have genetic factors that predispose to higher levels of some exposure, you can effectively randomize people to get higher versus lower levels of that exposure. And then you can say, just like in a randomized trial, is that exposure causally related to the outcome? So that's what we did, looking at depression and getting genetic data from large-scale depression studies, and also looking at physical activity in something called the UK Biobank, which is a very large study of 500,000 individuals, where there was objectively measured physical activity measured by accelerometer, the kind of thing that you might wear in a Fitbit or something, and subjectively also by self-report. And so what we're doing here is testing, is some exposure, you can see this little diagram here, an exposure X causally affecting an outcome by effectively randomizing people to higher or lower levels of exposure X based on their genetics. And then you can do that in 2 directions. So you can look at, does physical activity have a causal effect on depression? Does depression have a causal effect on physical activity? So just to get to the answer was there was a causal effect of physical activity on depression, that is lowering risk of depression. So for every 1 standard deviation increase in average acceleration, a measure of physical activity, there was an odds ratio of 0.74, so less than 1 protective. It was not seen in the other direction. So we didn't see a causal effect of depression on physical activity. And interestingly, we only saw these effects using objectively measured physical activity rather than asking people how active they were. Another example of how you can use genetics to actually study valuable interventions is in that area of social support that I mentioned. And again, led by Carmel, we've been looking in collaboration with the Army STARS project at resilience. And in this study, the Army STARS project is the Army study to assess risk and resilience in service members. And it included a cohort of soldiers who were studied before and after deployment to combat. And then we could ask over the next 9 months or so, who developed new onset depression. And we were interested in whether something called unit cohesion, which is a measure of how much the soldiers in a unit are connected, how much social support there is, how much do people have your back. And what we found was that if you create a genetic risk score for depression, a polygenic risk score, there is a stepwise increase in risk of depression. So if you're looking at that figure on the right in the A panel, where you see those 3 bars, that's low, intermediate, and high genetic risk. You see an increasing odds ratio for risk of depression after combat deployment. So that's an effect of the genetics. And then there was also a protective effect of unit cohesion. So you see that the more soldiers reported that sense of unit cohesion, the more protected they were from new onset depression. But here was the sort of hopeful, even more hopeful part of that, and that is that unit cohesion seemed to be a relatively robust protective factor, because whether or not you had low, intermediate, or high genetic risk scores, you still see that significant protective effect. And even whether you have low, intermediate, or high deployment stress scores. So this is just an example of how we can actually use genetics to study something like protective factors for depression or anxiety or other kinds of things. There are lots of large-scale studies that are now underway that are going to vastly increase our ability to learn these kinds of things. And one of them is the UK Biobank I mentioned before. There's the Million Veteran Program that has been conducted by the VA, and the All of Us Research Program, a national initiative now underway to enroll 1 million or more Americans, reflecting the full diversity of the country, and to really learn about, to advance precision medicine, learn about better ways to diagnose and treat and prevent diseases of all kinds, and to better understand healthy aging and other kinds of outcomes. So this is actually a pretty exciting time, and we are finally getting the tools. But just to bring you back to the final takeaways, the things I would have you remember is that, yes, genetic variation contributes to pretty much all mental health conditions that have been studied. We have actually begun to identify genes that are involved in pretty convincing ways, and that is providing new leads for developing treatments. Psychiatric conditions, depression, anxiety, and others are polygenic. It's not a single gene kind of situation. There are hundreds or thousands of DNA variants that each may contribute a small effect and may need to interact with the environment to show their effect. The genes are not one-to-one in terms of their influence on conditions. And in fact, there is a lot of genetic overlap between conditions that we think of as clinically distinct, which I think tells us something about a mismatch of our clinical classification and maybe the underlying biology that may teach us something new as we further refine our system of classification. Thus far, genetic testing is very limited. I gave you a couple of instances where it might be indicated, but so far limited, including the need for much better information about when pharmacogenetic testing might be useful. And finally, that ultimately genomic research may improve our ability to identify risk and resilience factors and really, we hope, change the situation, transform things from being more of a trial and error, one size fits all approach to something more useful, individualized, and precise. But it is not going to be an easy journey. So I will end there and be delighted to take questions. Thanks so much.
All right. Thank you very much, Dr. Smoller. We will now answer any questions about today's presentation. As a reminder, you can still submit questions through the chat box in your attendee control panel. So I actually have one question that I received during your presentation. Dr. Smoller, thank you for a great talk that covered information not often covered for clinicians. My question is about Fragile X premutation. I have had two different clients this year in my practice who have learned they are carriers. What is the clinical implication for their treatment? And do you have a paper to suggest I read?
Hmm. That's a great question. So Fragile X is a common cause of intellectual disability uh, in boys. It turns out that there are a number of studies that have implicated what's called the premutation. That is, um, a Fragile X involves a repeat of DNA sequence, and the longer it gets, the more likely it is to show an effect. But those who typically women who carry this premutation have been found to have higher degrees of social anxiety in particular, but other kinds of anxiety and perhaps depression as well. I think this is a good example of, and I think that was the question, right? It was about the premutation. Um,
Yes.
so I think it's a really good example of a couple things. One is um, that we are increasingly seeing genetics, in clinical practice and questions that come up. We didn't even talk much about direct-to-consumer genetics, which allows folks to get genetic information without going through a clinician. And so people are increasingly getting these kinds of questions from patients and clients uh, brought to your office without necessarily ordering a test. Not, I would say, and this is, to my knowledge, not much is known specifically about um, anything regarding the management of individuals with premutation Fragile X, the management of anxiety in that case, except to know that there may be a higher risk. To my knowledge, there is no reason to think that the typical treatments for anxiety would be more or less effective in somebody with a premutation. And so uh, I think, so another thing that it sort of underscores is there hasn't been a lot of research that has looked at when you have recognized genetic conditions, to what degree is the treatment altered. We have seen in certain conditions things that I mentioned before, copy number variations in schizophrenia, where it does look like the treatment that we would normally use in the absence of that for treating psychotic illness is essentially effective to the same degree, and that the symptoms are not easily distinguished as well.
All right.
And in terms of a paper, sorry, to follow up or something to
Oh, sorry,
read,
no problem.
yeah, I would be happy to. There are a couple things I can suggest, and I can send them to whoever would like afterwards.
Okay, I will connect the both of you. We have another question. In terms of protective factors for depression, do we know the difference between protecting against ever having an episode of depression versus protecting against subsequent episodes?
We don't, is the bottom line. We actually don't know a whole lot um, about effective prevention strategies. As I mentioned before, we know some general risk factors for incident, that is new onset, but also recurrent depression. But they are these things that we don't have a whole lot of. They're not highly actionable. Things like early trauma, and I mean, obviously, there's actionability to prevent trauma, which is a societal, tragedy. But by the time folks come in for treatment of depression or are seen, it's often the case that that damage has been done, and genetics the same way. In terms of things that I mentioned, like physical activity, social support, we don't have fine-grained information about the difference of incident versus recurrent depression, unfortunately.
All right, we have another question. Dr. Smoller, thank you for a fascinating and illuminating presentation. How can we address concerns that knowing one's genetic risk may intensify stigma and blame? Does the focus on genetic determinants of resilience make the most sense?
It's a great question, and it's one that has increasingly come up because, as I said, well, two things have made this even more of a live issue. That is, what does it mean when people find genetic information that might be related to illness? One is, in medical settings, at least, there is increasing movement towards implementing and incorporating genetics into care. And sometimes you get incidental findings. So even if you're not trying to test for some genetic basis of psychiatric disorder, something related to that might be found incidentally and returned to people because it has other health implications. The other thing, of course, is the direct-to-consumer genomics that people are increasingly engaged in and excited about, the ability to send off a sample and get your genetic information back. And there's a lot of back and forth and interest in, is it going to be destigmatizing, essentially, to better understand how genes may be contributing to illness? If we think back at 50 or 60 years ago, genetic factors were very much downplayed, and illness was typically blamed on poor mothering, and there was a tremendous stigma and burden. And the hope was that overturning that would really change things. There have actually been a number of studies now that have suggested that it's sort of a double-edged sword. For example, in studies of legal settings where people have asked for reduced sentencing or exoneration based on a genetic predisposition to a certain kind of behavior, and you ask people about, is that a mitigating factor? People sometimes think it is, but then they also feel like they also have somewhat more stigmatizing views in other domains of folks who may have a genetic explanation. So it is something for us really to think about. I think a key thing is education, because the fact is that, for example, in the depression paper that I showed you, in the abstract of that paper, right up front, they say, variation to, I forget exactly the words, but essentially it was, genetic contributions to depression are part of being human, essentially. We all have them to varying degrees. This is really also true in most other things, autism, related traits, and so on. So, part of it is education. Part of it is understanding that genes are not destiny. I do think this focus on resilience and prevention is one way of really helping people to understand the potential value of understanding these things. We've seen in studies of returning ApoE4, the risk factor for Alzheimer's disease, that people worried that in a condition like that, where there wasn't necessarily that much you could do about reducing your risk, which may be true in psychiatric disorder as well, at least so far, that people would really have bad psychological reactions, would trigger depression and maybe suicidality and so on. But that hasn't seemed to be the case in the studies that have looked at this. So it is a complicated issue, one that we really have to address more head on, I think, in the future years. It's a great question.
Thank you, Dr. Smoller. I will give participants another minute to think of any last-minute questions. All right, I think that's all for our Q&A. Thank you again, Dr. Smoller, for giving that great presentation, and thank you to our participants for attending today's training. You will receive a follow-up email shortly that includes a link to the webinar evaluation, webinar recording, and post-test assessment. Please stay on the webinar platform if you would like to take the post-test assessment after these few announcements.